All right, hi everyone. Welcome to part 3 of today's lesson on meiosis phases. So again, we're going to be continuing on with the coloring sheet, but this time we're going to be doing steps 4 through 6, and this time you actually get to color. Yay. So again, we are using the colors pink and blue to represent the maternal and paternal DNA. My recommendation is once we color this first cell together, I would then color the rest, like focus on just using pink and then switch over to blue. So I'll show you kind of what I mean. Otherwise it does get confusing on which ones you color. So we are going to start with prophase 1. So remember, this parent cell, this germ cell in the testes, has already gone through interphase. Okay, so it's made copies of its DNA, and at this point, the DNA has coiled and condensed itself into chromosomes, which we see here, and there are 6 of them. So let's go ahead and talk about like what's happening first, and then we'll do the coloring. So what is happening in this phase here? So the chromosomes, you'll notice, are matching up. They're pairs. This is what we call homologous pair. So let's go ahead and like label this one here so that we know what this is showing us. So I just did kind of like a bracket here. I'm going to write homologous pair. Okay, so that's just one pair, second pair, third pair. Okay, so what's going on in prophase 1 is that the chromosomes match to their homologous pair. Now, there's a reason why they're doing this. What they're going to be doing is these pairs, so again, they're going to be swapping parts of their DNA. Okay, so remember, chromosomes are just coiled, condensed DNA molecules. So they're going to be swapping their DNA, and that is what we call crossing over. Okay, so crossing over happens in prophase 1, and this is what's going on. So chromosomes will swap. And I'm going to say genes because they don't do all of their DNA. It's just like parts of it. So swap genes. And this is going to lead to genetic diversity. Okay, so remember, our whole goal of meiosis, again, is to make gamete cells that have half of the number of chromosomes, so they're haploid. We already covered that. But we also want to make sure that they're genetically unique, okay? So there is a reason for this. We don't want all of mom's DNA or all of dad's DNA. We don't like a swapping of it. So let's see what that looks like using the markers. So I'm going to start with pink first. And for every homologous pair, you want 1 maternal chromosome and 1 paternal chromosome, okay? So this one I'm going to be coloring pink. But here's what I'm going to do that's different, okay? So this was like a completely pink chromosome, but I'm going to pretend that it crossed over with the paternal DNA, which is blue. So I'm going to pretend that it swapped this arm and leg with the paternal chromosome. So I'm only going to cover in like half of that pink. And I'm going to switch over to blue and color in this arm and this leg blue. So basically like chunks, way to word it, basically get swapped. So let me draw the other one so you can kind of see what it looks like. So this one here, I'm drawing the paternal chromosome. So see how it's mostly blue, but it swapped part of its arm and part of its leg with that homologous pair. okay? And it's random every time. Maybe some homologous pairs never swap. They may not, and some do. So what I'm going to do here is actually color in the rest of these homologous pairs. The medium-sized one, I'm going to pretend that they don't cross over. So here's my maternal chromosome, all pink. And then this one over here is the paternal chromosome. Okay, so let's pretend these two contain the gene for eye color. So like mom's chromosome contains her eye color gene, and then dad's chromosome contains his eye color gene. Okay, and we're going to be making gametes, and eventually we're going to decide which one we want to put in each gamete. All right, let's do the smaller ones. This one, I'm just going to only have them swap their arm part. So let's go ahead and color those in. Notice I leave that part blank because I'm going to color it in pink. Okay. And then this one's blue. And then pink. All right. So that is prophase 1. Again, the big thing that happens, which I'm going to like make it bold here. So I'm going to put it in a box. Crossing over. That's the big thing that happens in prophase 1 is chromosomes that are homologous pairs are swapping sections of their DNA. And the whole goal again is for genetic diversity. Okay, so what you're going to do next is color the rest of these chromosomes. I'm going to color this one with you so that we can start to see like how they're going to pair apart, and then I'll kind of go through how I colored them so that you get a chance to catch up. Okay, so here we can see the homologous pairs are still lining up. We got our big pair, medium pair, and then small pair. In metaphase 1, these chromosomes don't like to like always stay like this is the blue side, this is the pink side. They can trade places. And again, that all comes back to we want genetic diversity. So on this one, I am going to draw most of the pink ones here. For this middle one, I'm going to have those two flip sides. So let me quickly draw this and then I'll show you what I mean. All right, so here's what I did. I colored in my chromosomes. Notice that I did match, so that big homologous chromosome pair basically are the same ones here, but now you're seeing them here for metaphase 1. Remember, metaphase 1 is where the chromosomes are going to line up in the middle, which is kind of what you see going on here. We'll talk about what's happening a little bit later. The medium pair that you see over here, they didn't do any crossing over, and that's totally fine. But you see them over here. Notice though, the blue chromosome is here on the right, and the pink one is here on the left. Chromosomes don't have to stay on the same side of their cell. They can trade places. And again, that all has to do with genetic diversity. And then on here, you can see my smaller pair. They match just the ones that we saw here. They only swapped basically the top part of their arms, which is what you can see down here. Okay, so what I want you to do is go ahead and pause the video, and you are going to color in the rest of these chromosomes, okay? Give me a chance to color in mine, and then I will show you what they look like, and you can kind of color as you go. All right, welcome back. So what we're gonna be doing right now is kind of briefly going through how I colored these. Feel free at any point to pause the video and you can color yours the same as well. So I'm just gonna start here at the top. Remember, crossing over has happened. This is where the homologous pairs are swapping their chromosomes. In metaphase, these pairs are lining up in the middle. So you can see here that I can see that crossing over. This one has like part of the maternal DNA, but it has an arm and a leg that contains the paternal because they swapped. These two did not, but they did trade places. And then these two did like a little mini crossing over where just one of the arms has traded. So I copied that same pattern as I went through here. And as you can see through meiosis too. So I'll kind of just go a little bit slow. Feel free to pause at any point so that you can copy down how I colored these. And then here's how I ended it. So you can see already, remember, we're trying to get so that each gamete is genetically unique. Notice that all 4 of these cells are not identical. They're actually genetically different from each other, and that is all because of this crossing over. All right.